like to thank you all for attending this talk and giving me your time. Good afternoon. My name is William Bowie. I'm a PhD student at Baylor College of Medicine, and today I'll be presenting some of our work on multiview factorization for joint modeling of spatial multi-omics and histology images via NMF. Biological mechanisms governing systems and tissues are inherently complex, necessitating comprehensive views across the genome, transcriptome, epigenome, proteome, and metabolome. Recent advances in single-cell technologies have enabled the simultaneous capture of DNA, RNA, and protein data at an unprecedented resolution. These advances have paved the way for significant discoveries, such as the identification of novel cell phenotypes in mice, prognostic biomarkers in pancreatic cancer, and the development of new tissue atlases. By integrating multiple omic layers, we can gain deeper insights into cellular functions. While this technology is very exciting, single-cell analysis do have an inherent limitation. Spatial organization is essential for understanding cellular processes, since cell communication and microenvironmental influences are inherently spatial. Spatial technologies are invaluable for elucidating cell communication pathways, understanding tissue disorganization and diseases, mapping molecular features across tissue boundaries, and performing comparative gene analysis between differing tissue regions. Spatial technology has been constantly improving, and much work has been done to extend spatial technology to other modalities of data, allowing for simultaneous profiling of multiple omics and accompanying imaging on serial sections of tissues, with spatial localization for all these serial slices to look at many different omic features on the same cell population and relative location. There exists a number of sequencing-based and image-based spatial technologies, each with their own advantages and disadvantages, capturing different numbers of genes or features at differing resolutions, capture sizes, and with differing quantities of cells able to be captured. This is summarized in the figure to the right. And here I point out spatial ATAC-seq and spatial metabolomics as two extra omic modalities that have extended to the spatial space, with more continuing to be developed. Given the exciting advancing field, there is a need to develop a computational tool capable of simultaneously analyzing and integrating the many omic layers we're able to capture from one tissue sample, along with the accompanying spatial and image data, and integrate them. For this, we look towards successful path methods, which solve a previous problem, integrating different single-cell omic studies across different samples, many of which notably use NMF as a basis. NMF, or non-negative matrix factorization, is a dimension reduction technique that imposes a non-negative constraint on data in order to reproduce factorizations that intuitively create a sparse and easily interpretable parts-based representation of data. To the right, I give the classic example of a face being decomposed into its parts through NMF, and this parts-based, easily interpretable representations can be extended to bioinformatic applications. Here, I highlight some well-known methods, such as LIGER, which uses NMF to decompose factors from shared features in different collected single-cell omics from different populations, and its extension, uni-NMF, which can decompose both shared and unshared features. In coupled NMF, the features not need explicitly to be shared, but connected through a with a coupling matrix. We hope to extend upon this great work and use NMF to focus on a different problem, integrating different omics collected from the same population paired spatially. To this aim, we propose a method that is very flexible in the analysis of multi-omics, and we first focus on the case of integrating morphology images routinely captured in spatial studies but often underutilized as a complementary modality in spatial transcriptomics and spatial ATAC-seq. Now, I'll go over our method, multi-NMF extra, in 3 steps, focusing first on inputs and preparing our matrix representations, then the underlying algorithm, and the output and downstream analysis. So, first, demonstrating the example of spatial transcriptomic analysis captured with vZoom, we perform standard normalization on the gene count matrix as the transcriptome representation for our data, but we must represent morphology with a descriptive matrix as well, so that they can be code decomposed. To do this, we take the H&E image and crop it, creating image tiles, where each image tile is cropped at the same spatial location registered using the spatial information from the STVZM side, so that each image tile represents the morphological information of that paired spot for which we have gene expression information. We then take each tile and send it through the pre-trained AlexNet CNN, and we pull, notably from the second to last layer, so that we can vectorize each morphology image into a vector that is rich in information and high dimensional. Aggregating these together allows us to create a final paired morphology feature matrix. Next, we incorporate the multiview NMF objective and strategy proposed by Liu et al. Now that we have both modalities or views of data represented within matrices, we first decompose each view into U basis or weight matrices and V component or factor matrices. Then we normalize each basis or weight matrix using L1 normalization such that they become comparable, and we calculate V consensus, which is the aggregation or shared information between both V component matrices. Looking closer at the objective function, we can appreciate that it's simply minimizing the difference between each view's original matrix X and its reconstruction, U product of V, and then minimizing as well the difference between each view's V component matrix and the consensus, but view-wise consensus between both views. This minimizes the disagreement between view-wise NMF components in both expression and the image space during optimization for a soft regularization towards a shared low-dimensional clustering structure. Taking our learned V consensus, we can plot them spatially and see that NMF reveals descriptive components which intuitively represent distinct areas where features from both views coincide. From these components, we also implemented a method to create a rank-based spot-to-spot similarity matrix, creating one per multi-NMF extra run in order to create an ensemble clustering approach which aggregates multiple runs from multiple initializations together before performing k-means, also with a high number of initializations for a spatial clustering that is highly robust, with the option to further smooth spatial domains with HRMF. To demonstrate a practical application of multi-NMF extra, we analyzed human samples of TNBC and ER-positive breast cancer collected from Wu et al. In a publicly available study. For this talk, I would like to focus on the TNBC sample highlighted in red. Here, I will demonstrate how multi-NMF extra drives interesting biological analysis with its ability to integrate rich morphological information with spatial transcriptomic data. Using the methodology previously went over, we can see that multi-NMF produces interpretable components, which we here show 20, and this gives a spatial clustering, which reveals transcriptionally and morphologically agreeable tumor regions, capturing many of the striking and interesting striations in the tissue captured through the H&E. Looking closer at the 20 structures we generated and illustrating each cluster spatially, we can see some interesting clusters have been discovered, specifically 3, 9, and 11, which are adjacent tumor and stroma clusters, with cluster 11 directly interfacing 3 and 9. We can see this here looking a bit closer in on the zoomed in image and demonstrate that our tool can outline populations and morphologically interesting tissue structures where we expect interactions of cells to be beneficial to study. Additionally, we show that multi-NMF's high agreeability with tissue structure allows you to narrow your focus down on these clusters and look into their biological relevance. We perform DE differential expression analysis followed by gene set enrichment analysis on several clusters, which we show here, and find GO terms enriched for blood vessel development. In cluster 12, T cell activation, 16, keratinization, and 6, epithelial mesenchymal transition. Given that this patient contains a lymphocyte cluster, cluster 12, which we've shown previously, we next ask whether T cells and B cells are spatially colocalized, indicated by the expression of CD8 and MS4A1 and our key marker genes. We see markings for T cells and B cells respectively, confirming their colocalization and providing evidence of a tertiary lymphoid structure. tertiary lymphoid structure is a morphological structure that has been shown to be associated with favorable prognosis. This successfully demonstrates our tool's ability to delineate important morphology-associated tissue features. Cluster 16 was often missed by other methods, but discovered by our own in analysis I'll show next. This is significant as this cluster is defined by the expression of LUCAT1 and SERPINE1, LUCAT1 being lung cancer-associated transcript 1, and a literature search revealing that this represents a novel cell proliferation and cancer invasion marker. SERPINE1 is a novel immune regulator. As a very recent paper shows, SERPINE1 expression promotes CD8 T cell exclusion. To verify this, we confirm that the cluster is devoid of CD8 T cells, and thus show analysis of integrative and morphology and expression clustering identifies immunoregulatory markers. We compared our tool to 4 other methods that are used to find spatial domains, given spatial transcriptomic data. Notably, 2 of these methods do not use image information, but SPAGCN and MOFA are capable of. We highlight differences of the methods in 4 morphological regions indicated by the black outlines, notably in regions 1 and 3, which are here. We see that other methods miss this distinct morphological feature in cluster. This is the cluster 16 that I just pointed out earlier, where we found immunoregulatory stromal markers. This highlights the importance of incorporating morphological images in describing the tumor architecture, benefiting over incorporating spatial information alone. Next, we saw unbiased measures of image similarity to objectively evaluate the results of multi-NMF extra. We tested 4 image measures, but for here, I'll focus in on FSIM. And we see that multi-NMF extra exhibits better performance to SPAGCN, the other method designed to incorporate histology information from H&E, measuring performance in terms of better intra-cluster image similarity scores, averaged from the cross-comparison of all H&E image tiles sharing the same cluster ID identified from each method. The other similarity indices gave similar results. For additional evaluation, we benchmarked using the VZM human dorsal lateral prefrontal cortex dataset generated by Manner et al., a common benchmarking dataset. And we see with performances ranging from 0.32 to 0.5 in different tissues and case-selected components measured by ARI, multi-NMF achieves comparable or improved results compared to other competing methods. We next demonstrate the flexibility of our tool to extend to other modalities and perform analysis on spatial attack-seq data, which jointly considers attack-seq chromatin profiling with captured image and spatial information, showing that our method is highly flexible in general and able to extend to other spatial multi-omics. Here I show 3 replicate samples of HER2 breast cancer generated recently by Bowie et al. In a paper published in Nat Biotech. We used Maestro for processing and peak calling, and then paired this with imaging data represented by the nuclear antigen staining shown here using the workflow previously described. Our final NMF clustering results show notable correlation with HER2 lesions seen in the stained images, showing too that morphology can be successfully integrated with chromatin profiling. To understand how chromatin accessibility varies across spatial domains, we find differently accessible peaks among 10 of the multi-NMF extra clusters using DE-seq. We discovered almost 3,000 differential chromatin accessibility peaks, which encode for spatial regulation and are potential regulatory regions for the tumor microenvironment. We illustrate this here in the heat map, where each cluster gets a 600 base pair window, and we show the peak intensity signal centered along the physical peaks plotted along the top. We also demonstrate that these differential regions are reproducible across our replica size, with 50 to 60% of these differentially accessible regions being additionally differentially accessible across 3 successive slides. Next, we take a look at a few interesting regions. First, we look at CCR2, which plays an important role in regulating T cell responses, and in the upstream of CCR2, we discover a differentially expressed peak, which which has a higher intensity in 2 of the 10 clusters we've shown, and is well supported by additional genomic evidence coinciding with ENCODE cis-regulatory elements in the layered HC3K27AC track, which I've highlighted with arrows, indicating this is likely to be an enhancer element. This peak also aligns well with species conservation scores as measured by phylo. Similarly, we find a CD28 peak in the intergenic region of the facility of the CD28 gene, with striking differential peaks into the clusters, this being a part of a co-stimulation pathway for native T cell activation, and one for lipogen antigen 86, another peak proximal to a gene that regulates immune cells. Overall, these cis-regulatory regions provide possible explanations for how the components of the tumor microenvironment are formed and provide interesting results that could be further analyzed with CRISPR-Cas9 in the tumor environment to investigate their functions. Here, I just quickly provide some benchmarking results in which we made a C++ implementation that greatly improved the speed upon the multi-NMF. And in conclusion, I've shown that we're tackling a challenging problem integrating different spatial multi-omics, but multi-NMF Extra is able to find highly agreeable features between different modality views and is extremely flexible. Future works would include optimizing the update rules to utilize coordinate block descent for faster convergence and combining morphology, transcriptome, epigenome, and a 3-view omic analysis. I would like to thank my lab, everyone at the Zoo Lab Baylor College of Medicine, especially my PI, Chen Zhu, and for funding CPRIT and the Department of Defense. Thank you so much for your time, and I'm now open for questions.
Thank you very much for a great talk. Any questions? Okay, I'll start with one question. So the factors that you found look really interesting and that they're really well defined. I'm wondering if you could, do you think you could extend that in the same way that multi-omics factor analysis did to do like compare different factors that come from different modalities, but for your breast cancer case to compare disease and normal? Have you ever thought about looking at factors that are normal or disease?
Yes, in the future, we definitely have plans to see if this tool can be utilized for vertical and horizontal integration across time samples, across different populations. I think that looking at these features, you can kind of see which ones are correlated together, and that can definitely be a useful or interesting area of study in the future. Thank you for your question.